All right, thank you very much, Paul, for the introduction. And everyone can hear me fine? So, yeah, like Paul said, before I get into the actual work that I do, I will give a little bit of the background of our center. What I particularly do is spectroscopy, and I'm interested in neurological diseases, and the work is essentially biomarker and surrogate marker work. So I'm at the CMRR on the other side of the river, and we focus on high, I don't know if you can see this, but high and ultra-high field MR scanning, MRI and MRS. And high fields start at 3 Tesla by definition, and ultra-high fields at 7 Tesla. The reason why we focus on high and ultra-high field MRI is it gives us higher sensitivity, higher resolution. So just little bit of background on how the center actually got started. The high field endeavor, in fact, started in early 90s, and industry did some experimentation. The first high field scanners that were built were actually 4 Teslas. Right now, everyone knows about 3T and 7T. Not so many 4 Teslas around, but it did start with 4 Teslas, and there were 3 around the world. And each of these manufacturers, the major MR scanner manufacturers, Siemens, GE, and Philips, had one as far as I know. And this is the kind of image they got at 4 Tesla, really blurry and not much resolution. So there was disappointment. And they decided to actually get rid of these scanners, and they ended up in academia. And one of them, the one from Siemens, was purchased by our center director and his colleagues. And they worked on the methodology. They did a lot of optimization. They had the physics background. And this is the kind of image they were able to get at the same magnetic field from actually the same scanner. So this was back in 91. So that's how it started. And they got the attention of both academia and industry. And then next, one of the first accomplishments was in functional imaging. You might have heard of BOLD fMRI, blood oxygenation level dependent fMRI, where people study brain activation upon stimulation. The first studies were done with visual stimulation, where the subjects see a pattern like this that's flickering, and their occipital cortex lights up. So this was first accomplished in a couple of labs, one of which was in the lab of Dr. Urbil. And this is how he looked like then, and this is how he looks like now. So, and based on that success and other successes that demonstrated the benefits of high fields and ultra-high fields, the field took off. Okay, maybe this doesn't work so well. So then, in 99, the first 7 Tesla was installed right here at the University of Minnesota. This is our center co-director, Mike Garwood, sitting in the bore of that scanner. This is how it was delivered. And there was that one in 99, and there are now over 50 either installed or on order around the world. So the field is really following, and a lot of major universities, they all have 3 Teslas now, and now everybody's trying to get 7 Teslas. Just wanted to show you our current setup. We received an expansion a couple of years ago. The left-hand side, maybe I'll use this. This part was our old building, and we got an expansion, a new wing here and a second floor. And so just to show you our scanners, this isn't working so well. Okay, I'll use these. So we have currently 5 machines for human scanning, 2 7 Tesla scanners, 2 3 Tesla scanners, and 1 4 Tesla scanner. That's been really our workhorse. It's not the very original one. It's a replacement, but that's been producing a lot of good work for us. And these are our animal scanners. So we have a 9.4 Tesla here, a wider bore 9.4 Tesla that's actually used for large animal scanning as well as human head scanning. And we have a 16.4 Tesla. That's the highest field available for rodent animal scanning in the world right now. And we are expecting soon a delivery of a 10.5 Tesla scanner for whole body human scanning. We're expecting, hopefully, in November, it's up to field. And again, that's going to be the highest field available in the world for whole body human scanning. So we have a lot of nice toys to play with, bottom line. And just to give you a flavor of what you can do with MRI, everyone knows about anatomic imaging. You can get really exquisite contrast and get nice anatomic images in the body. But you can also image perfusion, blood flow. You can image function, fMRI. You can image biochemistry, both metabolite levels in the body that are concentrated enough, as well as metabolic rates and connectivity. And there are many other ways to use the MR scanner to get different aspects of pathologies, for example. And this is just an example of anatomic imaging. And what you're imaging here is water. The body is 70 to 80% water. So, and depending on the immediate environment of the water molecules, you get different relaxation of your signal, which gives rise to this contrast. This is an example from our 7 Tesla, for example, to image blood vessels, magnetic resonance angiography. And this is at very high resolution, 0.4 millimeter isotropic resolution. My colleagues at the center produce such images. You get very nice tissue to blood contrast, as well as nice spatial resolution when you use a 7 Tesla scanner. This is another example, diffusion MRI. You might have heard of DTI. There's a lot of interest in this method, especially with the Connectome initiative to map the connectivity in the human brain. So what you're measuring, what you're determining here is essentially the wiring in the brain. And these are all the tracts. You map the axonal tracts in the white matter with this kind of technique. In functional MRI, it's dependent on stimulating a certain part of the brain. This is the back of the head. This image was acquired with a surface coil that the subject lies on for best sensitivity to see the back of the head and the visual cortex. When the brain is stimulated, blood flow to that area increases, deoxyhemoglobin levels go down with the increase in blood flow, and the fMRI signal goes up. So this is very basic description of this technique. And finally, this is the technique that I've been focusing on, spectroscopy. And we acquire neurochemical profiles of endogenous chemicals that are concentrated enough, 1 millimolar or higher, essentially, in the brain tissue. And this is the kind of type of data that we acquire. It's called a spectrum. Each of these peaks is coming from a different chemical. So we are looking at protons in these neurochemicals, and depending on their electronic environment, they give rise to a signal at a different frequency. So each of these chemicals essentially has a signature, and we acquire this spectrum. We know which chemicals contribute to this spectrum. We do a deconvolution type analysis, and we get millimolar concentrations for neurochemicals such as these. For example, N-acetyl aspartate, this big peak comes from NAA, and that's one of the main biomarkers that people have been working with. And that chemical in the adult brain is exclusively localized in neurons. So if you have some kind of neuronal dysfunction or loss, this peak starts going down. Creatine is this other big peak here. It's creatine and phosphocreatine. These are energy metabolites. So if you have some issue with energy metabolism, you might see alterations in that peak. Choline-containing compounds are constituents of membranes. And for example, if there is increased membrane turnover or demyelination, you might see this peak go up. And others. So at 1.5 Tesla, the standard clinical scanner, although that's changing now, everyone's trying to switch to 3 Tesla, but 1.5 Tesla has been the standard clinical scanner field. At that field, you're more or less limited to NAA, creatine, and choline. And depending on the acquisition parameters, you might get a couple of other neurochemicals. By going to 3 Tesla, 7 Tesla, and higher, the list of neurochemicals that you can reliably quantify increases, and you start being able to reliably quantify neurochemicals like glutamate, GABA, important neurotransmitters, or glutathione, vitamin C, antioxidants, other energy metabolism-related neurochemicals, etc. So just a little demonstration of what high fields give you. This is one of the chemicals that we quantify, myoinositol. It's generally considered a gliosis marker. If there is reactive astrocytosis or any other type of gliosis, myoinositol may increase, but it's also an osmolyte, so it has multiple roles. At 1.5 Tesla, this is the type of spectrum you get from this chemical. You go to 9.4 Tesla, where a lot of our animal rodent scanning happens. This is the fine structure you can obtain. So there is a lot more information to do a reliable quantification for this neurochemical. And for example, the labels refer to the individual protons. This signal over here comes from this particular proton. This one comes from this particular proton. It all depends on their immediate electronic environment. So a quick and I would say incomplete overview of the cellular and biochemical processes that one can possibly assess by MRS. Changes in cell number, viability, I already mentioned NAA as a neuronal marker. Glutamate in neurodegeneration, for example, tends to go down together with NAA, probably because most of the neurons we have are glutamatergic. Then we can possibly assess changes in glia using these chemicals, myoinositol and glutamine. Glutamine is primarily localized in glial cells, while glutamate is primarily localized in neurons, so they can be markers for those two cell types. Demyelination, increased membrane turnover, can lead to changes in choline-containing compounds and mobile lipid peaks that we also get with this technique. Defects in energy metabolism, we quantify creatine, phosphocreatine, lactate, and glucose. In oxidative stress, we quantify the two most concentrated antioxidant compounds, glutathione and vitamin C. And when thinking about this technique, you really need to be cognizant of where in the brain you're studying, because the neurochemical profiles are region-specific, as one would expect, because the tissue composition is quite different from different parts of the brain. Here I have examples of spectra from regions of interest for various neurological diseases, such as posterior cingulate. There's a lot of interest in that region for Alzheimer's and Parkinson's. White matter, putamen, substantia nigra, the brainstem pons, cerebellar vermis. And if you look at the patterns of these spectra, you're going to see, notice at least that the big peaks and their ratios to each other are quite different. For example, look at the ratio of this peak to this one, choline to creatine, which is very different than this peak. And when you acquire a spectrum from pons, it will always look like this, at least in the healthy brain. So this is a normal pons spectrum, and it's very region specific. This is showing quantitatively that there are statistically significant differences in neurochemical concentrations in these different regions that we studied in that work. This is a profile of chemicals that we can reliably quantify in all these regions, and you see there's quite a bit of variation. For example, look at glutamate. There is large variation between white matter and gray matter, for example. And so coming to my own work, I try to establish biomarkers in neurodegenerative diseases. The goal is to identify and validate these, and they can be utilized for various purposes: to monitor pathology, for diagnosis, early detection of neuronal dysfunction, as well as increased understanding of disease processes, to monitor disease progression and disease staging. And I think most importantly, to see drug effects, to be able to monitor disease reversal, and trying to establish objective outcome measures for preclinical and clinical trials. So as you probably know, neurodegenerative diseases have a very slow progression and a lot of phenotypic variability, and they require long clinical trials with large sample sizes, which are not always possible, especially in rare diseases. And on top of that, clinical outcome measures that we always need to utilize in the clinic as well as part of any study, they don't distinguish symptomatic versus disease-modifying drug effects. So for those reasons, I've been focusing a lot on this set of hereditary conditions called spinocerebellar ataxias, SCAs. They cause neurodegeneration in the cerebellum and in a lot of cases also the brainstem. Here I have T2 weighted images from a control volunteer and from a patient who has SCA1. This was the first gene discovered in the SCA series, hence the number 1, actually by investigators here at the University of Minnesota, Dr. Harry Orr being one of them. And you can see, this is the cerebellum, this is the brainstem, and you see the hyperintensity in the images, which reflects the cerebrospinal fluid around these tissues. So there is atrophy in this disease of the cerebellum as well as the brainstem. And that leads to progressive loss of motor coordination, and it affects gaze, speech, gait, and balance. So there are more than 20 genetic subtypes of SCAs, which provides you with a gold standard to test the sensitivity and the specificity of any biomarker that you want to establish. And in addition, there are faithful mouse models that were developed for these diseases that enable the studies of pathological correlates of any MR markers that you want to establish. So this is one of the studies that we published back in 2010. And a lot of the work that I will show will be in SCA1, spinocerebellar ataxia type 1. So here I have a cerebellum from this region of interest in a control volunteer and a patient with SCA1. And if you look at the spectral pattern, it is quite different in the patient versus control. This neuronal marker NAA went way down. The creatine peak is up. Myoinositol, the possible gliosis marker, is up, and glutamate is down. So you can look at an individual spectrum in one patient and distinguish patient from control. And that was a small study, 9 patients and 11 controls. And we had a number of statistically significant differences in the neurochemical profile. And recently we translated those methods that were developed on the 4 Tesla to our 3 Tesla clinical scanner, and using all the standard commercial hardware. And we were able to replicate the results in a larger patient and control cohort, actually. So the hardware was different, scanner as well as coil, the pulse sequence, the way we acquired the data was different, the operator as well as the patient pool was different. So this is internal replication of the results, which was good. And I like looking at these neurochemical concentrations in these 2D plots. In this case, we are looking at total NAA versus myelostol, the neuronal versus the glial marker. And these are the patients, these are the controls. And you see separation by using just two neurochemicals from the profile, separation between the groups with no overlap. This patient is marked here because that patient also had the highest ataxia severity score, the worst clinical status, so indicating a correlation between clinical status and these numbers. So this kind of relationship we replicated in our recent 3 Tesla studies. So these are data shown from the PONS, one of the several regions that we scanned, 3 regions to be specific. And you'll see there is one patient here that's overlapping with the controls. Actually, we had 2 pre-symptomatic individuals. So these are gene confirmed. They will definitely get SCA1, but they are not showing any symptoms yet. You look at the neurochemical levels. One of these 2 patients, also neurochemistry-wise, is not showing any signs of disease. While the other patient, even though there are no symptoms, there are already neurochemical indications that the disease has started. So this is pre-symptomatic detection of disease, and it's quite exciting. So we would like to get to early detection of these diseases. And this shows the correlation of at least one of the neurochemicals, the neuronal marker, again from the old 4 Tesla data in the PONS, with the clinical scale. This is a standardized, validated ataxia scale for the assessment and rating of ataxia. It goes from 0, no motor deficit essentially, up to 40. 40 is the worst score with the highest motor deficit. So we had early to moderate stage patients in this study. And it shows you the correlation between the clinical status and the neurochemical level. The worse the clinical status, the higher numbers, the lower the level of the neuronal marker. And this is the replication of this outcome at our more recent 3 Tesla study. And I actually plotted the controls and patients in separate colors here. And these are the 2 pre-symptomatic patients that I mentioned. So one of them really has NAA levels in the middle of the controls. The other one has NAA levels lower than all the controls we have in the study. So this reflects that the MRS markers really do indeed reflect the clinical status in these diseases. And it's also consistent with this notion that has been out there for some time now. This is a plot from this paper in 2003 that alterations, biochemical and otherwise, precede the symptoms of disease as well as structural changes in neurodegeneration. So you have a long period of time, years, where neuronal function degrades, and only at this point you start seeing the symptoms, and diagnosis comes even after that. And by that point, you might have lost half of your neuronal function already. So the goal is to catch people early on in this period with these biochemical measurements. So switching gears to some mouse model work, we are doing these studies in parallel in mouse models and patients. And just this is some background. SCA1 is dominantly inherited and late onset. It's due to an expansion of a CAG triplet repeat in the affected gene, and it results in a polyglutamine expansion in the ataxin-1 protein. And the first model that I will show you has an 82 polyglutamine expansion in ataxin-1. They overexpress, actually. This is a transgenic model. They overexpress mutant human ataxin-1 only in their Purkinje cells. So this is targeted expression. And they have been shown to reproduce the Purkinje cell pathology that's been seen in patients, as well as the phenotype. They develop progressive ataxia, as shown in this figure from it's an old paper from Harry Orr's lab. It was developed some time ago. So this is end-stage disease. They lose their coordination and balance. So the first question, of course, is if the neurochemical alterations we see in mouse models are similar to those that we see in patients. So here I have patient data, healthy control, SCA1 patient. Here I have mouse data acquired at 9.4 Tesla, wild-type mouse versus SCA1 mouse. And I have been talking about N-acetyl aspartate going down, glutamate going down, myoinositol going up. And we see those same changes in the mouse model as well. The advantage of the mouse model is, of course, that we can do some histology and look at the pathological correlates of these chemical changes. So, we are looking at the cerebellum in this case. You have a slice from a wild-type mouse with the molecular layer of the cerebellum, Purkinje layer, and the granular layer. And it's from this part of the cerebellum. And these are slices from mice with SCA1. This was a longitudinal study that went up to 1 year. So these mice were sacrificed, a subset of the mice were sacrificed at each of these time points. And if you look at the molecular layer, it gets thinner and thinner over time. So this is the atrophy, the cerebellar atrophy that we also see in humans. Looking at some MRS and MRI data, so this is again done with a surface coil that's sitting right on top of the cerebellum here. And this box is where we get the spectroscopic data. In the wild-type mouse, so this is one mouse scanned 4 times in their lifetime. And you can see not much is happening. The spectral quality is the same. We get identical spectra over time. You look at the SCA1 mouse. First of all, in the images, at 6 weeks of age, it looks like a normal size cerebellum, no atrophy, nothing. But over time, you see the shrinking, especially on the posterior side of the cerebellum. So we made the volume of interest, the voxel, smaller and smaller so that we would be scanning the same brain region. And if you also look at the spectra, you can, even in this one individual mouse, you can see the going down of NAA peak over time, the going down of the glutamate peak, and the increase of the myoinositol here over time. This shows the quantitative, semi-quantitative pathology scores or measures versus the neurochemical levels. Again, with NAA, myoinositol, and glutamate being the 3 neurochemicals of interest, versus the molecular layer thickness. These are data combined from all the mice that we sacrificed at different times. And you can see the neuronal markers, the thicker the molecular layer, these are more the control animals, the higher the NAA and glutamate levels. And myoinositol goes the other way because that does increase with the atrophy and with the disease. So there's nice correlations between the MR measures and the pathology measures. These are the same neurochemicals and showing their correlations with the severity score that my collaborator, Dr. Brent Clark, came up with. It's a score that goes from 0 to 4, 4 being the worst pathological state. And you see similar correlations with this semi-quantitative severity score. Looking at the 2D plots between selected neurochemicals at different ages, 6 weeks, 12 weeks, 24, and 1 year, we have the ataxic mice, the 82 polyglutamine mice here in the black symbols here. And we had actually two control groups. The SCA130Q is a positive control that overexpresses the normal human protein without the polyglutamine expansion, just to make sure that any changes that we detect are due to the polyglutamine expansion rather than the genetic manipulation of these mice. And you can see, starting at 6 weeks, there's absolutely no overlap between the groups. And this is the pre-symptomatic stage. These mice, at this point, are not ataxic yet. And the separation continues over time. And the numbers of mice, of course, goes down as we go through the study, because at each time point, we were sacrificing some to do the pathology on. So the second model is, I think, a very interesting model. It's a conditional model where my collaborators, Dr. Orr and his colleagues, put the transgene, the mutant ataxin-1, under doxycycline control. So you can feed these animals doxycycline, the simple antibiotic, and suppress the transgene expression, and they get better. So they were shown, the Orr lab has shown that the neurological phenotype, as well as the pathology, was reversible at early and mid stages when you treated the animals with doxycycline, and even to some extent at late stages. So we did a similar study using MRS to see if we would be able to detect this reversal also using our MR technology. We had two groups, early stage and mid stage. And the treated group, the doxycycline treated group in the early stage, received treatment from 6 to 12 weeks. So during that time, their transgene was suppressed. And the mid-stage group received the treatment from 12 to 24 weeks. And we had an untreated group, and we had the background controls. And this is looking at a treated mouse at the bottom and not treated mouse on top. Again, visually, this is at the beginning of the treatment, before treatment we scan them, and at the end of the treatment. You can, again, just by looking at the spectra, you can see a small but visible treatment effect. For example, NAA goes down, in the treated animal, and myelostol goes down. So these are normalization of these neurochemical levels. And this is looking at both groups and neurochemicals that did show a response to treatment. They were, again, NAA, myelostol, and taurine. It's a different osmolyte, and we don't know why this one responded, but it was actually completely normalized. So I have the three simples, the wild types here in the open squares, and treated and non-treated animals here, the treated ones in the triangles, and they are, with treatment, approaching the wild type levels in terms of their neurochemical concentrations. The reversal was not complete, it was partial. You're seeing, except in this case and in this case, NAA and myelostol only come maybe halfway to normal levels, and actually that's what histology also showed. It wasn't a complete reversal of the pathology. So we had similar nice correlations between the MR measures and histology measures. In the last animal model, we looked at this Knockin model. It has a longer polyglutamine insert, but it's a Knockin. So the 154 CAG repeats were introduced into the mouse, the endogenous SCA1 locus. So in this case, the expression of the bad protein essentially is everywhere in the body, and not only Purkinje cells. And interestingly, probably because they are not overexpressing this bad protein, the cerebellar pathology is much milder than the transgenic line we studied, which was good for us because we were able to look at very early pathology. So these are images from a wild-type mouse and from a Knockin mouse. At end stage, these animals actually don't even live as long as the transgenic line. This is 39 weeks, and you don't see any alterations in structural imaging, no indication of atrophy. And when you look at pathology also, there is no overt pathology even at end stage. And these are the quantitative pathology measures, molecular layer thickness over the 6-week to 39-week period. There's really no difference. Looking at the severity score, this is the same scale going from 0 to 4. Only at end stage we were able to pick up at 39 weeks a half-point difference in the severity score. But even in this case, the severity score of 1 indicates some pathology in the posterior lobule, not even covered by our MRS voxel. So in the voxel that we are looking at, there really isn't overt pathology. However, we did see quite significant neurochemical alterations starting at 6 weeks. So these 4 chemicals, creatine, the energy metabolite, choline, might be indicative of various processes. Taurine is an osmolyte, glutamine, the glial neurochemical, and also an osmolyte. They changed over time, starting at 6 weeks. So switching gears to Parkinson's disease, it is characterized by degeneration of nigrostriatal dopaminergic neurons, which gives rise to motor symptoms. So motor neurodegenerative disease, motor disorders have been a theme in my investigations. But in the last several years, people have been discovering or showing some data that the caudal brainstem might even be involved in the disease process earlier than the nigrostriatal involvement. And this involvement actually causes some non-motor symptoms, sleep, anxiety, and depression. So this was based on a lot of detailed pathology work by Brock and his colleagues. And the idea is that the disease starts in the medulla here and then moves up to pons and to upper levels. And there was one prior spectroscopy study before we did this investigation of the brainstem. And this was a 3 Tesla study, but they only quantified 3 neurochemicals ratios, Na to creatine, and they saw no differences in patients versus controls. So we decided to do a 7 Tesla study, looked at 3 different regions. Pons was of high interest, and then putamen striatum is very much of interest for Parkinson's disease, and substantia nigra. And of all the neurochemicals that we are able to quantify, GABA showed a difference in both the pons and the putamen. And the difference between patients and controls in the pons was actually larger percentage-wise than the striatum, which would be consistent with an earlier involvement of this region. And going back and looking at literature, actually people had shown elevated GABA levels in the striatum, especially in the putamen, post-mortem in extraction studies. But this was never shown. And the reasons for a GABA elevation can be many. There's a lot of GABAergic regulation of neurons that project to the striatum from the pons, like neurons in the locus coeruleus, etc. So there's probably some involvement in the pathogenesis of the disease, higher GABA tone in that region. Then I talked about monitoring treatment response. So this was a pilot study that we did recently in Parkinson's disease as well as Gaucher disease. Gaucher is a lysosomal storage disease, and there has been a lot of interest in a mechanistic link between Gaucher and Parkinson's because people who have this hereditary lysosomal storage disease, Gaucher, are at a higher risk for developing Parkinson's disease. So we studied them side by side, looking at effects of N-acetylcysteine on cerebral glutathione levels. Now, N-acetylcysteine as an antioxidant received also a lot of interest recently because it's the precursor for this main antioxidant we have in the body, glutathione, and it's actually available as an oral supplement and in a variety of diseases where oxidative stress is considered as a major player. People are trying N-acetylcysteine, but nobody has shown if it gets into the brain and if it actually affects glutathione levels in the brain, and that's what we wanted to do. So this was a really little pilot study, 3 patients with early to moderate Parkinson's disease, 3 patients with Gaucher, and 3 healthy controls. They received a baseline MRI scan, and then we took them out of the scanner. We started an IV infusion of N-acetylcysteine, a large dose, and midway, that was a 1-hour-long infusion, and midway through the infusion, we placed them back in the scanner and started measuring glutathione, or continued measuring glutathione. These are actually the raw data of both brain glutathione in these black circles. With the baseline scan here, this is the period where we took out the patient and then placed them back in the scanner to continue measuring glutathione. And we have the blood reduced over oxidized glutathione ratios. So this is a good indicator of oxidative status in these volunteers. And the N-acetylcysteine infusion starts at time 0 and goes until 60 minutes. And so these are the 3 patients with PD, these are the 3 patients with Gaucher, and these are healthy controls. And you can see in each case the increase of the blood GSH to GSSG ratio, it increases, and once the infusion stops, it starts going down. In each volunteer, we saw the glutathione levels increase. You can follow these time courses, and you can detect the change in the time course, the elevation of the levels, and there is a lag relative to the blood levels, which you would expect with any infusion. There's always a 20 to 40 minute lag between blood and brain levels of any compounds infused. So they start going up, and they kind of level off, but we had to stop the study here because volunteers already have been in the scanner for a long time. So this is just a proof of principle showing that we can affect antioxidant levels in the brain by infusing a precursor. Next step, of course, is to try oral dosing of N-acetylcysteine because IV infusions, at least in the case of Parkinson's and Gaucher, would not be practical. And finally, a pediatric disease at the end of my talk. This is actually an old study. This was my first clinical study, I have to say, with collaborators from the Department of Pediatrics, adrenoleukodystrophy. It's a hereditary condition, and it's due to it's a metabolic problem. It's due to an impaired capacity for beta oxidation of very long-chain fatty acids that leads to their accumulation. And there are several forms. The childhood onset cerebral form is the worst type, and it involves inflammatory demyelination of the white matter. Bone marrow transplantation is a promising treatment, but needs to be performed early. So one of the important questions is who is going to get the cerebral form or not? And if they are going to get it, let's do a BMT. And in structural imaging, this is a T2 weighted image, you see lesions in the white matter that look like this. So in this patient, the disease started posteriorly. To show you some spectra from patients, these are actually both patients who have ALD. One is from normal appearing white matter here, and one from a lesion area, and the spectral pattern is completely different. You can see the N-acetyl aspartate It's way down. Glutamate is down. Glutamine is up. Myoinositol is way up. And this is lactates and lipids. We actually start seeing mobile lipids in this disease, which is interesting because it is a lipid storage disorder. So very clear differences in lesions. But even before lesions, maybe we have a chance to detect some differences. So this is one patient that we followed over 2 years, a baseline scan, 1 year later and 2 years later, and always from the same volume of interest in the frontal white matter here. At this point, there is really not much change in the image in this volume of interest, and not much. This looks like a normal spectrum for that brain region. 1 year later, still not much going on. The lesion expanded from the posterior part of the white matter, but still has not reached, on the structural image, the frontal white matter. But if you look at the spectrum, it's already a full-blown lesion based on the chemical profile. Lipids and lactates have increased. NAA has gone way down. Glutamate has gone down. It's more difficult to see, but in quantification, when you get the concentrations, these are clear changes. Glutamine has gone up. Myoinositol has gone up. And these changes are progressive 2 years later, 2 years after the baseline too. And at that point, you can see the lesion on structural imaging too. So this is confirming the ability to detect early chemical changes. And a subset of the patients we actually monitored before and after, up to 2.5 years after bone marrow transplantation. So these were 6 patients, and these were the 2 neurochemicals that most clearly responded to the treatment. One was glutamine, the other one was lipids plus lactate, so the mobile lipids, it looks like were going down. And these different symbols are individual patients, and you can see the response in each patient in these patients with ALD. So you can monitor the treatment response in individuals. So in summary, I have shown you that MRS detects parallel neurochemical alterations in patients and mouse models. That was from the SCA work. I have shown you that the same neurochemicals, in particular NA, myoinositol, and glutamate, reflect the clinical status and pathological progression. Again, the example here was SCA. Neurochemical alterations can be detected prior to symptoms and prior to overt pathology. I have shown the examples from SCA. And prior to structural MRI changes, that example was from ALD. And treatment effects are detectable. And in all the diseases that I have shown examples from, this was possible. And that's, I think, really the most critical piece for the long-term utility for this technique. In my acknowledgements, I have had wonderful collaborations, collaborators along the way, and most of these are still active collaborators in various departments, including pediatrics. Elsa Shapiro and Larry Charnas were my collaborators on the ALD project. And my colleagues from CMRR who contributed in various ways to the data that I have shown you. And this is a picture of our ataxia clinic, Dr. Bushara, the ataxia clinic director, who is my clinical collaborator on the ataxia work, our study coordinator, and funding resources. And thank you for your attention.
Yeah, I got one to start. So can you do spectroscopy on other tissues as well?
You can. You can.
On liver or kidney or those types of things too?
Yeah, I mean, in the body, you have to deal with motion effects. They are much more prominent with breathing. The head is easier from that perspective, but it is doable. So it affects your spectral quality. But I have colleagues at the center who do spectroscopy in the prostate, for example, prostate cancer applications and breast breast cancer applications, Pat Bolan and Greg. So yes, you can do it. Or muscle, you can do, sure.
What are you going to do with a 10 Tesla magnet?
What are we going to do? First, we need to get it operational, and that takes a long time, actually, months until everything is calibrated and ready to go. And then we're going to, I guess, try to push the limits of detectability. In our case, for neurochemicals, in the case of various imaging modalities, spatial resolution is really a big deal for them, how much SNR gain is. But the higher you go in magnetic field, the more the engineering challenges, and so you have to do a lot more technique development and optimization, because things that work easily at 3 Tesla may not work so well at 7 Tesla, so you have to overcome some technical issues. But once you overcome them, you can start applying.
Questions?
Raghu.
Question is regarding the functional MRIs. Where is the field right now?
Functional MRS? Well, functional MRS is, like, just for background, like a bold fMRI study. You do, for example, visual stimulation, and you look at the neurochemical response. And I have colleagues at the center who have done some very nice work in that area, showing the immediate chemical response to, for example, heavy visual stimulus. What those colleagues are now interested in, I mean, I just know locally, are the inhibitory responses rather than just excitatory, so GABA instead of glutamate. So, but I mean, in terms of where is the field, there have been a lot of groups around the world who replicated the results that came out of our 7 Tesla scanner with the neurochemical profile changes in response to visual stimuli. So that was good. But I think people are done with that now, so it's time to ask some new questions. But I haven't seen a lot of that.
I have one more too. So, I mean, it's really interesting that you get a metabolic sort of snapshot of what's going on in various tissues. Can you predict impending apoptosis and necrosis? I mean, can you tell which areas of the brain are metabolically impaired and ready to die?
Well, I have not focused specifically on that, I mean, apoptosis question, but I would guess yes. And we wouldn't be necessarily detecting a chemical that's in the pathway of the apoptotic switch. But if the metabolic processes are resulting in alterations in any of the profile of neurochemicals we are detecting, which I would expect, lactate, NAA, those would be affected. NAA has been shown to reflect mitochondrial dysfunction. And actually, in our mouse model work, too, the cell death comes at the end of life. The changes we see, the most dramatic changes we see, are way before the cell loss. And at the cell loss stage, NAA, mannose changes actually had leveled off. So we do really see that prior biochemical snapshot. But I would say the metabolites are not necessarily the primary metabolites that are in the pathway of whatever is happening at the cellular level, but secondary.
Okay. Any other questions? All right. Thanks.
Thank you.